Welcome to Biochemistry DSP channel. Today's topic will be about high metabolism of high density lipoproteins, HDL metabolism. High density lipoproteins helps in the transport of cholesterol from peripheral tissues to the liver. The major apoproteins in HDL, that is high density lipoproteins, are apoprotein A1, and it also have some apoproteins A2, apoprotein C, and apoprotein E. It serves as a plasma reservoir of apoprotein C and apoprotein E, which can be transferred to VLDL and chylomicrons. So, when you consider the metabolism of HDL, this HDL takes part in both lipoprotein triacylglycerol as well as cholesterol metabolism. It is synthesized and secreted from both liver and intestine. And HDL has the highest density among all lipoproteins. It is composed of 60% of lipids and 40% proteins. The lipid components will include mostly it will include phospholipids and 25% of cholesterol and small amount of 5% of TG. Okay. The major function of this HDL is to act as a repository for the apoprotein C and apoprotein E required for the metabolism of chylomicrons and very low density lipoprotein VLDL. Nascent Hdl, that is a newly synthesized HDL, consists of discoid phospholipid bilayers which contains apoprotein A and free cholesterol. So the nascent HDL, it's also called as discoidal HDL, which consists of discoid phospholipid bilayers and free cholesterol. Mainly it is synthesized in the liver and is released in blood by exocytosis. Plasma HDL has opposite function to that of LDL. HDL removes free cholesterol from extrahepatic tissues with the help of the enzyme phosphatidylcholine cholesterol acyl transferase PCAT. This enzyme is also called as lecithin cholesterol acyl transferase LCAT, uh, which helps this lecithin cholesterol acyl transferase helps to transport cholesterol, okay, which is in the HDL to the liver, where cholesterol is catabolized for its excretion outside the body. Okay, so the role of this LCAT is it's an enzyme which catalyze the esterification of cholesterol to form cholesterol ester. So it when cholesterol reacts with lecithin, it forms lysolecithin and cholesterol ester with the help of the enzyme lecithin cholesterol acyl transferase. So with the help of this LCAT only, HDL removes the cholesterol, free cholesterol from the extrahepatic tissues, and it will transport it to the liver where it gets catabolized. That's why it's called HDL is called a good cholesterol. So this okay. Now we'll see the metabolism of this HDL. So this HDL is nascent HDL is synthesized in the liver, and nascent HDL, since it consists of a discoid phospholipid bilayer with apoprotein A and free cholesterol. It's since it's discoidal, uh, it can be called as discoidal HDL. Okay, it's also called nascent HDL or discoidal HDL. And these lipoproteins are similar to particles found in the plasma of patients with deficiency of enzyme lecithin cholesterol acyl transferase, and in the plasma of patients with obstructive jaundice. LCAT, okay, the LCAT activator is apoprotein A1. This LCAT activator, apoprotein A1, will bind to this disc, and the surface phospholipid and free cholesterol will be converted into cholesterol ester and lysolecithin. This here itself you can find this for binding of this LCAT. To bind this LCAT with this discoidal HDL, apoprotein A1 is helpful. So after the once apoprotein A1 activates LCAT, this LCAT will catalyze the hydrolysis of this free cholesterol. It will convert all the free cholesterol into lysolecithin and cholesterol ester. Then the cholesterol esters will move into the hydrophobic interior of the bilayer, and the lysolecithin will be transferred to plasma albumin. So a non-polar core will be generated, forming a spherical HDL. Okay, so this spherical HDL structure is called as HDL3. So so this is how we classify this HDL as HDL1, HDL2, HDL3. Okay, so the nascent cholesterol, okay, the nascent form of HDL, which have discoidal shape, is called HDL1 form. After the role of LCAT, most of the free cholesterol will be converted into esterified cholesterol. So the shape also will changes from the discoidal. So when the reaction continues, as the reaction continues, still HDL becomes spherical in shape with lot of cholesterol esters. So when this disc is loaded with lot full of cholesterol esters, this particle, this HDL particle, will be designated as HDL3. So this is HDL3. Uh, uh, in this way, HDL1 will be converted into HDL3. So this spherical HDL will be covered by a surface of polar lipids and apolipoproteins. So this HDL3 also will be covered by lipids and apolipoprotein A1. It will also have this apolipoprotein A1, as well as it will be bound with this LCAT. So LCAT system is involved here. Okay, LCAT system is involved in removing the excess of unesterified cholesterol from lipoproteins and tissues. In this way, okay, so in this way the HDL L3 also, with the help of this LCAT, it will also remove the excess of unesterified cholesterol from the lipoproteins and tissues. The receptor which is necessary for this HDL binding is the class B scavenger receptor B1. Okay, the receptor is named as SRB1. SRB1 is nothing but the class B scavenger receptor B1. It has been identified as the HDL receptor in the liver. So HDL binds to the receptor via apoprotein A1. So apoprotein A1 is necessary to bind with for the binding of HDL with this HDL receptor SRB1 in the liver, and cholesterol ester is selectively delivered to the tissues. The transport of cholesterol from the tissue to liver is called as reverse cholesterol transport and is mediated by HDL cycle. Okay, so this part, the transport of this cholesterol from tissues, it collects this HDL particle. Is role of HDL is to collect all the cholesterol from derived from extrahepatic tissues, and it will transport that cholesterol to the liver through this SRB1 receptor. It is called as HDL receptor. So this reverse cholesterol transport, okay, this is called as the reverse cholesterol transport and is mediated by HDL cycle. So here the smaller HDL3 will accept cholesterol from the tissues. Okay, so how this HDL3 will receive this cholesterol ester means with the help of ABC protein. So ABC protein is called ATP binding cassette transporter 1 protein. So this ABC ATP binding cassette transporter 1 is a member of family of transporter proteins that can couple the hydrolysis of ATP to the binding of a substrate. So it enables the transport, it will be enabled for the transport across the membrane. So after being accepted by HDL3, the cholesterol is then esterified again with the help of this LCAT, increasing the size of the particles to form the less dense HDL2. So from HDL3 you derive HDL2. The cycle is completed by reformation of HDL3 either after selective delivery of cholesterol ester to the liver via this SRB1 receptor or by the hydrolysis of HDL2 phospholipid and triacyl glycerol by the hepatic lipase. So either by these two ways the cycle is completed by reforming HDL3 once again. So in addition, the free apoprotein A1, okay, in addition, the free apoprotein A1 is available in the HDL2, which will be released, okay. The free apoprotein A1 is released by this process and it forms pre-beta HDL after associating with a minimum amount of phospholipid and cholesterol. It is called, after combining with the least amount of cholesterol and phospholipid, it will be called as pre-beta HDL. The pre-beta HDL is most the potent form of HDL in inducing cholesterol efflux from the tissues to form discoidal HDL. So again, it helps to synthesize this discoidal HDL. Then the HDL concentrations also vary reciprocally with plasma triacyl glycerol concentration and directly with the activity of lipoprotein lipase. Phospholipid and apoprotein A1 are released during the hydrolysis of chylomicrons and VLDL, which also will contribute to the formation of this pre-beta HDL and discoidal HDL. HDL2 concentration are inversely related to incidence of coronary atherosclerosis, possibly because they reflect the efficiency of reverse cholesterol transport. HDL1 is found in the blood in diet induced hypercholesterolemic animals. It is rich in cholesterol and its only apolipoprotein is ApoE. It appears that all plasma lipoproteins are interrelated components of one or more metabolic cycles that together are responsible for the complex process of plasma lipid transport. So this is about the metabolism of HDL. So as a summary, we get so this HDL is synthesized from liver. Uh, the newly synthesized nascent HDL is called discoidal HDL, which consists of discoid phospholipid bilayers and some free cholesterol, which will be released in blood by exocytosis. So the LCAT enzyme, which is present in this discoidal HDL, will help to transport free cholesterol from the extrahepatic tissues to liver, where it gets catabolized for its excretion. Such a transport is called as reverse transport of cholesterol. So after removing the free cholesterol from the extrahepatic tissue, the disc-shaped nascent HDL will be changed into spherical HDL, which is taken up by the liver through HDL receptor called SRB1 receptor mediated endocytosis. In liver, HDL cholesterol is catabolized to bile acids, or it can be secreted as free cholesterol in bile for its removal from the body. This plasma HDL also serves as a reservoir of apoproteins, ApoE and ApoC, that can be taken up by the nascent chylomicrons and VLDL from the circulating HDL. Since this HDL brings about reverse cholesterol transport, this HDL scavenges blood vessels from atherosclerosis, and also it will reduce the risk of coronary heart diseases. Therefore, a negative correlation exists between the incidence of coronary heart disease and plasma HDL cholesterol level. Higher the plasma HDL cholesterol means lesser will be the incidence of coronary heart disease. So this is the reason why we call HDL cholesterol as good cholesterol. So atherosclerosis and coronary heart disease are directly correlated with plasma LDL cholesterol, whereas it is inversely related with plasma HDL cholesterol. So so the functions of HDL is to scavenge. Okay, so HDL scavenges the extra cholesterol from the peripheral tissues by reverse cholesterol transport. It helps with the help of ApoE. This HDL, with the help of ApoE, competes with LDL for the binding sites on the membranes and prevents internalization of LDL cholesterol in the smooth cells of the arterial walls. It contributes its ApoC and ApoE to nascent VLDL and chylomicrons for receptor-mediated endocytosis. HDL stimulates prostacyclin synthesis by the endothelial cells, which prevents thrombus formation. HDL also helps in the removal of macrophages from the arterial walls. Thus, I hope you would have understood about the concepts of metabolism of high density lipoproteins as well as the functions of high density lipoproteins. Thank you for watching this video. Subscribe and support the channel Biochemistry DSP.